Okay, I'm actually ready now. Cell membrane, homeostasis, and cell transport. So we're going to talk about these 5 things. The 5 things we're talking about are structure and function of the cell membrane. We're going to talk about what homeostasis is. We're going to talk about what concentration gradients are, the difference between active and passive transport, and what it means to be hypertonic, hypotonic, or isotonic. So let's get going. Let's start off with structure and function of the cell membrane. Okay, so uh, there's a picture of the cell membrane. It's got a whole bunch of lines all over it, but I kind of want you to ignore all of the labels here. We're just going to focus on the main components of this that I want you to know about. And the main components of this are, first of all, um, let me put this in red so it's easier to see. But this cell membrane, this is your cell membrane. So this would be like outside of your cell. This would be like the inside of your cell. The membrane is just a barrier to separate the inside from the outside. Okay, so first let's talk about its structure. What is it made up of? And as far as the structure goes, the things that I want you to know that's important to know are, first of all, these things right here, which we call lipids. More specifically, they are actually called phospholipids. Okay, they're called phospholipids, and each one looks like a head with 2 little tails. And you can see them if you look really closely right here. You got the head with the 2 little black tails there. And if you also look closely, you'll see that there's actually not 1 layer, but 2 of these. So this, what we call this right here, is actually the lipid bilayer. So it's obviously bi means 2, so this is just 2 layers of lipids, and that's the way that it is built. We've got lipids here, and you'll also notice that the head and the tails of one, the tails of the other one face each other, and the heads face away from each other. Okay, so this is what creates the barrier between the outside of the cell and the inside of the cell. And also, so lipids make up the main part of the cell membrane, but also embedded in the lipid bilayer are other things like these guys, which are proteins. Okay, so these are what we call, they are proteins, but they are transport proteins. Okay. So these are transport proteins, and they do just what their name implies. They help things to transport into or out of the cell. Stuff needs to get into the cell, stuff needs to get out of the cell. This is how they get into and out of the cell. Actually, there's other ways that they can also get in and out, and we're going to talk about that here in just a second. Okay, so this cell membrane is what we call semi-permeable. And if you don't know what the word permeable means, the word permeable means it can be gone through. Okay, so like a brick wall is not permeable for you. Okay, you can't go through a brick wall, but you can go through, say, water, right? If there's like a waterfall, you can walk through it. That's something that's permeable. Okay. Permeable just means it can be gone through, but it's only semi-permeable, meaning that only some things can go through, but other things can't. And that's really important because, you know, when you're talking about your cells, you don't want just anything to be able to go in and out of your cell just whenever. You want to have some degree of control over what goes in the cell. You want to keep bad stuff out. You want to let good stuff in, right? So you don't want to be completely permeable, so that's why it's called being semi-permeable. And its job, the job of the cell membrane is to, first of all, give it structure as well as regulate what goes in and what goes out, like I was just saying, okay? And the main reason why it wants to control what goes in the cell and what comes out of the cell is because the cell membrane wants to maintain something that we call homeostasis. Okay, this is a really important thing that we're about to talk about. So, what is homeostasis? Well, let's find out. So, homeostasis is a stable internal environment. And when we say stable, what we mean is it's balanced, okay? So like I said, we have, our cell, we have stuff outside of the cell, and we have stuff inside of the cell, and we have this membrane, the cell membrane, which controls what goes in, what comes out, and the whole point is to maintain a stable internal environment within the cell. But what does it mean to be stable? Okay, so when we say stable, what we really mean is we're talking about stability in terms of concentration. Now, if you don't know what concentration is how much stuff there is in a given space. Okay, so imagine that you have a box, or let's just say this is a room. Let's say this is a room, and the room has people in it, okay? So each of these little dots is a person. Let's make them blue, just so that they stand out. Now, if all the people in the room are crammed in a corner, then that is not an even concentration, okay? You have a high concentration in a small amount of space, okay? So all the people are in this one corner here. That is a high concentration in that corner, but a low concentration in the rest of the place. Now, if we wanted to get to homeostasis or stability, what we would do is we would spread these people out evenly throughout the room. Okay, so every person would be evenly spaced from the other. That is stable. That's a balanced concentration throughout the room. Okay, and this is what your cells want, but they want the concentrations to be the same outside as inside. Now, this does not mean the same amount, okay? You could have, if you have water outside the cell, your cells want the same concentration of water inside the cell as outside the cell, but that doesn't mean that the same amount of water needs to be on both sides. If I took this cell and I dropped it in the ocean, there's obviously going to be more water in the ocean. We can't have all the water or half the water in the ocean go into the cell and have the same amount. So it's not about amount, it's about concentration. It's about the percentage of stuff that there is in a given space. This might make more sense here in just a second. Okay, so let's go on to talk about concentration gradients. Okay, so the next one, what are concentration gradients? Okay, so if we have stuff on the outside of the cell and we have stuff on the inside of the cell, the stuff on the outside, let's say that there's a high concentration or a high percentage of these red dots outside the cell versus the percentage of red dots inside the cell. Let's say there's only a couple, okay? So outside we have a high concentration of these red dots, whatever they are, it doesn't matter what they are, just there's a high concentration of them outside, and there is a low concentration of them inside, okay? This creates what we call a concentration gradient. And a concentration gradient always flows from high to low. Now, think of a river. You jump in the water, you can feel the current of the river, right? There is a current that is flowing from one place to another place, okay? So think of this concentration gradient as the flow of the river. Now, things naturally want to flow in that direction because the current of the river will take those things in the same direction as it is going. If you just throw something in there and you let it go, it'll go in the same direction as the concentration gradient, okay? But sometimes they need to move things in the opposite direction of that current. And we're going to get to that here in just a second. But I just want you to know what a concentration gradient is for now. And we're going to talk about the different kinds of things here in just a second. And so, like I said, a concentration gradient is just a difference in concentration from outside to inside or inside to outside. This could be high down here and this could be low outside the cell. So this currently is not in homeostasis, okay? The cell wants to be in homeostasis, meaning that it wants the same amount of stuff. Let's see, how many red dots do we have outside? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Let's do one more, okay, to make 12. So we want the concentration on both sides to be the same. So what needs to happen is if we have 8 out here and 4 down here, well then how many need to go inside in order for the concentrations to be the same? Well, 8 and 4 is 12. So to be the same on both sides, you'd need 6 on the outside and 6 on the inside, right? So we need 2 of these guys to go from outside to inside so we can have 6 on the inside, 6 on the outside. Now the concentrations are balanced. There is no high concentration, there is no low concentration. And now the concentrations are balanced on both sides, and we have now reached homeostasis. And this is what your cells want. This is what your cells are always trying to get, okay? But it doesn't always happen, and it's not always easy. And sometimes you have to make homeostasis happen. It doesn't just happen on its own, okay? So now let's go into what we call active and passive transport, okay? So in active transport, the difference between the two is, remember how I said that cells have a concentration gradient. If it's high outside and low inside, the concentration gradient is flowing from high to low or from outside to inside, okay? Now, as I said before, this is like the current of a river. If you throw something in the river, the current will take it in that direction that it's going in. This requires no energy. If you hop in the river, and you do nothing, you will just flow in the same direction as the gradient. This is what we call passive transport, okay? So passive transport requires no energy from the cell. It just happens on its own, okay? Active transport wait a second. So the reason why it doesn't require any energy is because we're going from high concentration to low concentration. And that requires no energy to make that happen, okay? However, active transport, it does require energy. And the reason it requires energy is because we are going from a low concentration to a high concentration. And this is like if you were to go in the river and you feel the current pushing against you, but you go upstream against the current. You have to put out energy to do that. So going from low to high requires energy. And you're probably asking, well, why would your cells do that? Don't they want balanced concentrations on both sides? Why don't they want to just let it flow naturally until it balances out on both sides? Well, the reason is because sometimes stuff needs to get out of the cell. Even if there's more of it outside the cell, like for instance here, if there's a lot of stuff outside the cell and there's not very much of it inside the cell, but your cell still needs to get whatever little bit is left in here out, it's got to go from low to high, meaning it's got to go against the concentration gradient, meaning it has to put out energy to make that happen. Okay? So sometimes you have to do active transport. You have to force it to happen. Okay? So there's different kinds of active transport. There's what we call endo and exocytosis. There are what we call pumps, and these require energy, which is ATP. Okay, so ATP is an energy molecule that you may not know what that is yet, but if you don't, just know that this is like if your cell was a car, this would be its gasoline. Okay, so ATP is the energy that your cells are going to use to pump things from a low concentration to a high concentration. Okay, so passive transport does not require energy. It goes from high to low, and this is also known as what we call diffusion. Okay, so diffusion is just another name for passive transport. So if we go back to the cell, remember how the cell membrane is made up of heads and tails, right? One head, two tails, one head, two tails. And these guys are side by side. But here's the thing about passive transport. Some things are small enough to go from outside the cell to inside the cell. Just simply because they're so tiny, they can squeeze right through the gaps. If not that there really are any gaps, but there are tiny little spaces between these two heads. They're not bonded together, okay? So something, if it's small enough, can just squeak right through. And the three things that can do that are water, oxygen, and CO2. That's supposed to be an ampersand, and I'm not very good at drawing them. There we go. That's better. H2O, oxygen, and CO2. Okay, so this could be H2O, oxygen, or CO2, and these things are small enough to simply just go in and out of the cell just by squeaking through the phospholipids in the cell membrane because they're tiny enough. Okay, and that's a form of diffusion because the cell doesn't have to exert any energy for that to happen. It just happens on its own, okay? They just flow with the current, whichever direction the current happens to be flowing at that time. When you have diffusion with water, this is what we call osmosis. So osmosis is diffusion with water. Okay, so when it's oxygen and CO2, that's just plain old diffusion, okay? But when it's diffusion with water, we call it osmosis. And we could just call it diffusion with water, but because it's so common and plays such a big role, you give it its own special name and we call it osmosis, okay? And we're going to talk more about that in just a second. But let's say that something needs to get from high concentration, like these water, oxygen, or CO2, they're small enough to go through the cell membrane because they're that tiny. But what if you're not that tiny and you still want to go from high concentration to a low concentration? But you're too small, or I'm sorry, you're too big to fit through the gaps in the cell membrane like these guys are, those three there. What do you do then? Well, then you actually go through one of the transport proteins that kind of looks like a hot dog bun if you cut it in half. And the transport protein is the thing that I showed you on the other slide at the very beginning. Now, this is still diffusion. It's still diffusion because we're going from a high concentration to a low concentration. And anytime you go high to low, that's passive, which is also diffusion, right? So it's still diffusion, but instead of it being a regular kind of diffusion like these guys do here because they're tiny enough, they have to go through basically a doorway in the cell membrane. So it's diffusion, but it's facilitated diffusion. So facilitated diffusion is diffusion with transport protein. Okay. All right. So again, passive transport doesn't require energy. High to low, also known as diffusion. Regular diffusion is stuff that's small enough to just squeak in and out of the cell through the cell membrane because they're tiny enough. That's water, oxygen, and CO2. Oxygen and CO2 is just normal diffusion. Diffusion with water we call osmosis. If you're bigger than these things, you're too big to just go in and out of the cell whenever you want. You have to go through a transport protein, which is like a doorway to a room, okay? You're too big to just squeak through the holes in the wall, so you have to come in through the door, and that's facilitated diffusion. Still diffusion, but just being helped or facilitated by a transport protein that's in the cell membrane, okay? Once back to active transport, it does require energy. It's whenever you want to go from a low concentration to a high concentration. There's these things called endo and exocytosis where they take massive things and literally have to push them out of the cell. If the cell were a room, it's like imagine if you had to get something in the room, but it's too big to go through the door. You would have to break down the wall to the room, roll the big giant thing in there, and then build the wall back. When you're going into the cell, it's called endocytosis, and when you're taking something huge out of the cell, it's exocytosis. So if this were a big giant thing that we need to get out of the cell, it's too big to go through the transport protein, so we literally have to break down the wall, move it out, and then once it's out here, build the wall back, build the cell membrane, not a wall, but the cell membrane. It's like the wall to a room, okay? I don't want y'all thinking I'm talking about a cell wall here. So anyway, that would be exocytosis. The opposite is endocytosis. If you were going into the cell, that would be endocytosis, okay? And you remember these transport proteins I was talking about? Well, when you are going from low to high, but you have to go through a transport protein, that transport protein is what we call a pump. It's just like if you were to pump something like air into a tire, there's already more air, a higher concentration of air in the tire than there is outside, but you got to force more in there anyway. You need a pump to do that. So these transport proteins turn into pumps whenever you have to go from low to high in either direction that it goes, okay? All right, so we talked about osmosis a second ago, about how that's just diffusion with water. So water is one of the things that just goes in and out of the cell all the time as it pleases. And so now I'm going to go to this next one, which is talking specifically about water going into and out of cells, specifically red blood cells. Okay. So there's what we call hypertonic, isotonic, and hypotonic. And basically, the difference between these three is which direction is water going? Is water going into the cell? Is water going out of the cell? Or is water not going into or out of the cell? Is it balanced on both sides? Well, hypertonic is where water leaves the cell. And think of the cell as a water balloon. If water goes out of the water balloon shrinks, right? So if water leaves the cell shrinks. That's hypertonic. The way to remember that is if you're hyper, you have a lot of energy, you run around a lot, you get smaller because you lose weight because you're running around a lot because you're hyper. Okay, so hyper, you shrink. Okay. Hypotonic, when you hear hypo, think hippo. And hippos are fat, right? Everyone knows hippos are fat. So just think hypotonic, the cell gets fatter. Why is the cell getting fatter? Well, that's because water is going into the cell, okay? And so just like a water balloon, when water goes into the cell, the balloon gets bigger, the cell gets bigger, okay? But if it gets too big, then it pops, right? Which is bad, so you don't want that too much. Isotonic is where it's balanced. The same amount of water coming in is the same amount of water leaving, and so the water balloon doesn't change size. The cell doesn't change size. It's stable. That's how it's supposed to be. Okay, so just remember, hyper, you lose weight because you're running around. Iso, you're staying the same. Hypo, hippo is fat. Cells get bigger, cells get fatter. All right, so in summary, on cell membrane structure and function, the cell membrane is made up of proteins, lipids, and carbs, all three of those things. It's semi-permeable. Some stuff can come in, some stuff can come out, but not everything can come in and come out. It's made up of a lipid bilayer, 2 layers of those lipids. It provides structure and regulates cell transport, and it maintains homeostasis. What is homeostasis? It's that stable internal environment. Think of Goldilocks, not too much, not too little, just right. And what does that mean though? To be just right, what it means is to have a balanced concentration on both sides of the membrane. Okay, so they want the same amount of stuff or the same concentration of stuff on the outside as the inside. If there's a difference in concentration between the outside and the inside of the cell, it creates what's called a gradient. The gradient could go from out to in or in to out, depending on where the high is and the low is. Remember, it always flows from high to low, and it acts like the current in a river. Okay, so whatever direction that current's going, that's the direction that stuff is going to want to go. And if you don't do anything, it'll go there. But if you want it to go in the opposite direction, you have to put out energy. That's where active and passive transport come in, okay? So if you're going against the concentration gradient, that needs energy. Passive does not. Energy is ATP. It's just another name for the energy molecule that your body uses, okay? So active transport uses ATP to pump stuff against the concentration gradient from low to high, if you need to do that. The way that things get into and out of the cell one of the ways is through these transport proteins, and in active transport they act like pumps to pump things from low to high against the current of the river or the concentration gradient. Endocytosis and exocytosis is for big stuff, so stuff that's too big to go through the door in the room. You have to break down the wall or basically tear apart the cell membrane, push the big thing out or in, depending on whatever you're trying to do, and then build the membrane back. Passive transport is also known as diffusion. Any type of passive transport is diffusion. There's regular diffusion with water, O2, and CO2. That's where they're tiny enough they can just go in and out through the cell membrane because they're so small they can fit through the gaps. Diffusion with water, we give it a special name called osmosis. And if it's too big to squeak through the cell membrane, it has to go through a transport protein. It's still diffusion because it's going high to low, but it's being facilitated with the transport protein. So it's called facilitated diffusion. And when we're talking about water going into or out of a cell, remember that hypertonic cells will shrink if they're in a hypertonic solution or a hypertonic environment. The cell will shrink. Hyper, use a lot of energy. Shrink because you're losing weight. Hypo means hippo. Hippo is fat, right? The cell swells in a hypotonic environment. Isotonic, it's just right. The cell is not changing size, bigger or smaller, because the amount of water on both sides is balanced. Water's not going anywhere. The cell stays the same size. All right, poof, you're smart.